Hello and welcome to the latest Technology Networks webinar, Powering AI-Based Protein Engineering with Scalable Data. I'm your moderator, Dr. Stephen Gibney, Science Writer for Technology Networks, and I'm excited to be here to host today's session. We have a fantastic presenter, Dr. Ryan Emerson, who will be sharing some of his valuable insights with us today. With over 10 years' experience in R&D leadership at the intersection of tech and biology, Ryan is passionate about applying data science to solve complex biomedical challenges and improve human health. As Senior Vice President of Data Science at Alpha Bio, he oversees the development and implementation of computational models and tools to support the discovery and optimization of novel therapeutics. As a General Manager of Platform Solutions, Ryan leads the delivery of Alpha's platform technologies to pharma and biotech partners to accelerate AI-powered drug discovery. After his presentation, there'll be a short Q&A session. We encourage you to submit your questions at any point during the presentation. To do so, just type your question in the box that's on the right-hand side of your screen and click send. We'll do our best to address as many questions as we can today with the time we've got available. During today's webinar, we'll be asking a quick poll question, which will appear on your screen, and we kindly ask you to respond when prompted. Answering the question is required in order to receive your certificate of attendance by email once the webinar comes to an end. If you encounter any technical difficulties during the webinar, just click the chat box on the right-hand side of your screen to request assistance from our support team. Without any more introduction, I'm now going to hand over to our speaker, Dr. Ryan Emerson. Ryan, over to you.
Thank you very much for the introduction, and thank you for joining me today to the audience. I'm excited to talk about how we power AI-based protein engineering with scalable data at Alpha. I'm going to start with a really brief intro to Alpha Bio. We bill ourselves as the protein-protein interaction company, working to improve human health by measuring, predicting, and engineering protein-protein interactions. So we originally spun out from David Baker's lab at the University of Washington's Institute for Protein Design. So kind of really the thought of marrying high-throughput biology to AI-based design is in the company's DNA. We are working with technology that was, in some sense, literally conceived to validate AI designs at scale and generate useful training data. We are a venture-funded startup still in Seattle, and we've got a team of a few dozen with widespread expertise in synthetic biology, protein engineering, and machine learning. And really, we work with our wet lab platform, AlphaSeq, which I'll describe in a little bit, which allows us to measure millions of protein-protein interaction affinities quantitatively. And AlphaBind, which is our term for the AI system kind of trained and then applied on AlphaSeq data, with over 1.8 billion protein-protein interactions measured to date. And we put those together to do advanced protein engineering, which I'll tell you about. I'm going to start with a general overview and a few examples of how AlphaSeq data can power AI protein design in a variety of different ways. And I'm going to finish in the second half of the talk with a more focused look at antibody optimization as one of our most common use cases, showing how we put AlphaSeq data together and our AlphaBind models to really transform the way we think about antibody engineering. So I'll start with an overview of our thoughts on how experimental data are really essential to power cutting-edge protein design. So AI workflows are increasingly popular. They're increasingly powerful. They're really super neat. And we think that there is a role to be played by high-throughput biological data at really every stage of the journey toward a functional AI protein model. So it starts with the generation of large-scale, diverse data to train our models. And in some sense, I think the industry has been kind of subsuming 30, 40 years of accumulated public sector data for a long time, but we're reaching the point for sure at which models have trained on all of the easily available data, and those databases are no longer growing at an exceptional rate. And so new sources of data are going to be required if we want to scale these models another 10, 100x with training data. We also do a lot of work with problem-specific data. So something I'll show you later in the antibody optimization section is that dedication with a specific molecule to generating high-throughput wet lab data really massively accelerates and enhances the impact of these AI models. So we do a lot of work generating data around particular molecules in order to fine-tune foundation models for specific tasks. And then more on the AI research side, we think there's a real need for benchmarking data. So testing model performance in a highly parallel way, prospectively. Anyone who's written a lot of computational papers knows it's very hard to tell what's actually working. And there's often a trade-off where the things that are easiest to do, like cross-validation in silico, are sometimes the least predictive of what happens when you go and try and apply these things in vitro. The most predictive of how a model is going to work live is to attempt the actual design task you care about in the long run, but that requires easy access to lab data at scale. And then finally, just validation data to screen designs. So models are getting better. I think, really within the recent few months, maybe 6 months, we've started to see models that are claiming success rates high enough that you might actually be able to get away with a few 96-well plates of designs. But even then, the best models today require the ability to generate many designs and then test them to see which ones work, because most of them won't. And on harder problems, or for models that are attempting more difficult things or less refined models, sometimes that hit rate is even lower, in the 1 in 1,000 or even 1 in a million range. And so just every part of every AI protein workflow eventually needs a downstream step where you go in vitro and make sure that the molecule does what you'd like it to do. And we really think a lot about how to apply our wet lab technology to each of these problems. And so I'm going to step back and just talk a little bit about antibodies. They're great for therapeutic purposes, but a really tough nut to crack for existing protein design models. So these models are again, data constrained in general. So, the entire PDB still looks like an extremely small source of training data. If you think about the kind of precedents that are set, say by big tech training English language models, right? The entire corpus of protein structures is just not as big as we'd like. And for certain therapeutically relevant molecules like antibodies, the training corpus is very, very small. There are only about 10,000 published structures for antibodies in complex with their targets. There are only about 1,000 for nanobodies or kind of single chain camelid antibodies in complex with their targets. And in addition to that, the underlying sources of these data are only growing a few % each year. So waiting around for people to crystallize more things is probably not going to be the way that we get a fundamental step change in model performance by feeding in more training data. And one consequence of this is that antibody antigen complexes are a particularly tough nut to crack for all of these models when you think about co-structure prediction. So here on the right, we've got a figure from Pissarra et al. Showing at .q scores, which is just kind of a measure of the co-complex quality when we try and fold an antigen and antibody structure together, and showing that performance is really quite poor across all of the cool models. So we've got AlphaFold 3 here, Chai, Boltz 2. Models are improving all the time, but just want to call out that antibodies in particular have been tough, and we think they're probably tough for a number of reasons. They're data constrained, and of course, you're trying to model kind of long, loopy CDRs, which are not the nice, tightly packed protein structures that are probably easier to access for these models. And so where do we come in? So we do a lot of work with AlphaSeq, which is the kind of core wet lab technology that got the company off the ground, invented in David Baker's lab. And this is a high throughput assay for quantitative measurement of protein-protein affinities. And so it is a two-sided yeast display platform where we will make barcoded libraries of antibodies or other binders and barcoded libraries of targets. We can mix those, and based on a next-gen sequencing-based readout of yeast fusion events, we actually create a sequenceable event that gives us a quantitative affinity measurement for every edge between the binder library and the target library. And so the results of this assay are effectively a rectangular matrix of protein-protein interaction affinities on the order of a few 100,000 to about 1 million protein-protein interactions at a time, with a quantitative range from just about sub-nanomolar to maybe low double-digit micromolar. And so we run this assay. We've generated to date a little less than 2 billion PPIs. And the rest of this morning, I'm going to be talking about how we apply that to accelerate protein AI models. And here I want to stop for a quick shout out to Twist. So most of the work that we do is based on Twist DNA libraries of one sort or another. So fundamental to AI protein modeling is the idea that we can make up a sequence that we'd like to test and then go test that in the lab. And to do that, we need fully defined DNA at a scale that was really not possible a decade ago. And so I want to briefly talk about some of the different DNA sources that I'm gonna go through today. So we use a lot of gene fragments. So to build highly diverse libraries, kind of native human proteins or scaffolds for variant libraries, we'll use gene fragments. We can get dozens to hundreds of these fully defined, arbitrary length. We are now doing a lot of work with multiplex gene fragments. So critically, these things have recently hit the market and can go out to about 500 bases, which for an antibody engineer is a really magical place, because that means we can now fully define either the light chain or the heavy chain of an antibody, or fully define a VHH, which again is really critical. As you'll see later, if we take a scaffold and we want to make 15 or 20 mutations, that's something where we need to be having fully specified DNA. And we also do a lot of work with oligo pools. So oligo pools are really affordable, and we can get up to hundreds of thousands of candidates. And for these, we'll do a lot of work with variant libraries, say where we're editing kind of CDR1 through CDR3 of a heavy chain, for example. You'll see some of that data today. Or most of a domain on a native human protein. Or if you're into mini proteins are great to work with for synthetic DNA, because you can get really arbitrarily large libraries of fully defined sequences with this product. And so that's really powering it. We get these fully defined DNA libraries, we put them into yeast, we get really high throughput affinity measurements out. And I'll go over in the next couple slides a few examples of how we've worked with partners to apply our wet lab assay to really accelerate their AI modeling, their AI validation, AI training, and really kind of become the lab in the loop for some of these models. And I'll start with a teaser for the second half of the talk, which is our AlphaBind model. So this is an example of training a model for antibody optimization. This has a MABS paper that goes with it that we published with NVIDIA earlier this year. The problem here is that traditional antibody optimization is slow, it's labor intensive. It generally requires several rounds of kind of guess and check editing to get to a molecule whose properties we really like. Our solution here was to take a sequence-based model and pre-train that with about 7 million antibody antigen affinity measurements from our AlphaSeq assay, and then use that as part of a lab-in-the-loop system that also includes fine-tuning on candidate-specific data sets to train an accurate affinity prediction model. And with this model here in the lower left, we're showing what can be achieved. So on the x-axis here, we're showing the number of mutations introduced into a parental antibody. And here, critically, we want to point out that it takes a few mutations generally to achieve the best affinity that we're going to get from a particular antibody targeting a particular epitope with its own binding mode. And we can reach that pretty quickly. And after that, what we're really doing with our engineering is accumulating degrees of freedom that we can then use to engineer other properties we'd like. So to humanize an antibody. to make an antibody as germline as possible or otherwise reduce predicted immunogenicity risk in order to optimize melting temperature or expression in a CHO system, or all of these other properties that we really care about in a drug-like molecule, require us to make targeted edits to that antibody. And the goal of our system is to be able to define as wide a radius as possible in primary sequence space around our initial hit, where we can accurately find high affinity binders. So on the y-axis, we're charting the proportion of our AI-derived designs that actually improve in affinity compared to the parental antibody hit that we started engineering. And so in gray, we've got data from a state-of-the-art protein language model alone. So this is ESM2, but with no fine-tuning data. And we show that we get a success rate of about 20% on single mutants, maybe 10% on double mutants, declining pretty rapidly. And I want to call out that that's actually fantastic. 5 years ago, you would have said that was spectacular performance with no other information. But if we pre-train a model with all of these data, if we commit to fine-tuning a model with candidate-specific information, we can really dramatically enhance the efficacy of these AI models and get to a point where we can now push out to about 20 mutations out. So that's something like 15% primary sequence divergence, similar to a typical kind of human to mouse level of conservation, and still get the average candidate a little better in affinity than the parental candidate. More on that in a few minutes. We also use fine-tuning critically on some of the hardest problems in biologics that have severe demands for binding multiple targets. So here I'll show a little bit of data about a partnership we did with Gilead on an infectious disease therapeutic, where the diversity and constant mutation of the pathogen between patients and within patients really limits the impact of some of these biologic drugs. And so the goal here is actually engineer something with broad cross-reactivity to hundreds or even thousands of known clinical variants. And we did that with iterative training and fine-tuning. So we had a starting molecule, we explored the sequence space around it, trained an AI model, made new candidates, iterated several times across that workflow, and were eventually able to generate AI-optimized biologics that showed 3 to 30-fold improvement in neutralizing titers. So this is even downstream of protein characterization. This is going back to a viral neutralization assay, and showing that on the right here, we've got 5 different viral strains, and we're looking at the AI-designed candidates as a swarm plot, and looking at the fold improvement in neutralization titer. And the candidates, the viral strains are ordered in terms of the relative potency of the parental biologic. So virus E was strongly neutralized by the parental candidate. And there we saw about a 3-fold improvement in neutralization potency. But for some of the viral strains that were least well engaged, least potent in the parental biologic, we saw up to a 30-fold improvement. And so here we're showing that when we have a really finicky problem, like binding multiple targets, the breadth of this data is really what the AI models need to converge on a sequence that is going to be efficacious. I mentioned that every AI protein model eventually needs downstream validation, and I think the better the scale, the happier we're going to be on that. So here's an example that we did in partnership with Dreamfold, which is a really cool AI first protein design company, using the assay to validate de novo mini binder designs at scale. So here you can read any number of papers where people will put their AI model to work and test 96 designs or a few hundred. Here, Dreamfold generated 10,000 designs for each of 6 different targets, and we ran those all in an AlphaSeq experiment, measuring both on-target affinity and off-target affinity across targets, allowing us to very rapidly validate which designs were sensitively and specifically binding the target that they were supposed to. And so here we've got some example data on the bottom right here, where we're simply looking at affinity to the intended target on the x-axis and affinity to the best unintended target on the y-axis. And here you can see a big swarm of points that don't bind anything. That's pretty typical for most AI protein models. But also a few points out here that bind their intended target, but also bind unintended targets. So we think largely these are kind of misfolded more than anything. But critically, we can rapidly identify in this setup which designs were efficacious and start moving those forward. So in this one experiment, we actually validated almost 1,000 de novo designs in one pass. Another critical thing, as I mentioned, is benchmarking AI models. And so here, in partnership with Google and the Bill and Melinda Gates Foundation, and this work is described in a preprint on bioRxiv, we did a great project benchmarking different AI models for antibody optimization in a SARS-CoV-2 context. So this was when the pandemic was really still raging. Looking at some starter antibodies, so in particular antibodies raised against SARS-CoV-1, and seeing if they could be engineered as potent and cross-reactive SARS-CoV-2 binders. And one critical application of the assay here was to actually do that model training and optimization a few different ways and directly and prospectively test which ML models were more effective by using the assay to test tens of thousands of designs across different AI models and then prospectively assess success rates. And so by doing this and over several rounds of iteration, the AI designed variants managed to achieve up to 200-fold increase in affinity compared to the baseline parental, and even got some that bound emergent strains that hadn't even been seen during training. So the Omicron strain in particular is one that really ablated binding to that parental biologic, but not the AI designed version. And again, all of this is powered by lab-in-the-loop data generation and Twist DNA libraries that allow us to get millions of affinity measurements at a time. All right, now I'm going to briefly talk about how we put AlphaSeq and AlphaBind together for one of the most common things we work on, which is antibody optimization. So again, here, the traditional path is kind of a winding and circuitous path where we start with an initial hit. We notice that it has properties we don't like. We go and try and fix them. Maybe something else breaks. We go back and try and fix that. Obviously this is an exaggeration, but this is maybe accurate 10, 15 years ago. But it's really a circuitous path to the drug-like molecule that we'd really like to have. What we can do in just a few months is really do rapid simultaneous optimization across just about everything that we care about, really by starting with a really effective affinity regression model to map the entire space of high binding antibodies and then be able to select in silico the ones we think are going to be well-behaved drugs. And so again, there are a lot of things that we're asking of these biologics. We're asking affinity, and in some sense, that's pretty easy. That's the closest to what evolution likes to use antibodies for. And so we'll often have a pretty good starting point there. We can immunize something, we can do phage panning, and we'll get a nice specific binder. But we ask a lot more out of a drug than nature asks out of many antibodies. So we would also like it to be developable. We want something that's going to be easy to manufacture. We often want something that's specific. So depending on the particular clinical thesis we're working on, we can want something that is, targeting a specific variant or targeting one paralog, but not the other, having differential effects across different binding partners. And in certain cases, we also want cross-reactivity. So, in infectious disease, we need that clinically. For almost any drug, we would love it to be strongly cross-reactive to macaque, for instance. Rodent, if that's possible, right? That really eases preclinical development. And so because we're asking a lot, we think it's really important to utilize these AI protein models to ask, how can we get the best molecule we can as quickly as possible? And here I'll show some more data from our AlphaBind process. Again, this is experimental validation showing that we can train an affinity model that goes out to 20 or more mutations from a parental antibody. So the workflow here is that we start with a parental antibody. We have our pre-trained model. It's been trained on high throughput affinity data. We then generate one data set for model training, which is about eight weeks in the lab with twist oligo libraries. We now have a fine-tuned affinity model for this candidate, which is going to be highly accurate. And now we can generate in silico really highly affine and very sequence diverse antibodies. Again, here we're looking at violin plots. This is a validation of model performance after that one round of data generation. The x-axis is the number of mutations we've introduced. On the y-axis is relative affinity from the parental. And as you can see, in the shaded bits, we've just colored how many candidates we've got. So it's very easy for us to generate in silico thousands of candidates with hugely diverse primary sequence that all have really excellent affinity. And then downstream of that, we apply all of the best AI models we have for developability to pick out the very small subset of these that we want to go test in the lab, but with enough sequence diversity to really get our pick of the litter from those developability models, and an accurate affinity model that ensures a majority of those are going to bind stronger than our parental. And so again, with this model, we can engineer really excellent antibodies in just one go around this lab in the loop optimization cycle. So on the bottom here, we've got an example from a recent campaign showing what we can do with this system. So from the millions of high affinity antibodies we can generate in silico, we then screen based on predicted developability, take a small number into the lab, and end up with a molecule that's really excellent in a number of ways. So here we're showing CHO titer, purity by size exclusion, and a DSF assay for thermostability. And the red dots on the left are the lead candidate we started with. It's kind of in an acceptable range for all of these properties, but not great. And then starred, we've got our new lead. So we applied this AI system, we trained the affinity oracle, we filtered very harshly for predicted developability, and plated out just 24 of these to actually express as protein, and found a candidate with dramatically improved CHO titer, better purity by size exclusion, and really significantly improved thermostability. And so the idea here is that if we map out the space of antibodies with excellent affinity and have some variants to pick from, and we can then screen those in silico for ones we think are going to be well-behaved drugs, we can find those really rare antibodies with both excellent affinity and really good developability properties. So in the end, what we do is combine wet lab and dry lab technologies to deliver high quality data and high quality drug candidates. So again, our input is designed sequences, and this is why we interface so well with modern AI protein models. We then have our lab in the loop for generating data, for benchmarking models, for actually doing antibody optimization. And we output millions of affinity data points, which we can use to engineer really excellent therapeutics in just a few months. And with that, I want to thank you for your attention. Thank the entire team at A-alpha. And we've got contact information here if anyone wants to get in touch. Thanks very much.
High-throughput screening is fundamental to biomedical research, particularly for the development of protein-based therapeutics. By iteratively screening protein variants, researchers explore a protein's vast sequence space in search of a novel variant with desired, optimized properties. However, finding the right protein variant is difficult, even with the latest AI tools. Large-scale screens and training datasets for machine learning require pools of DNA, encoding thousands of defined protein variants to effectively sample the protein sequence space. There are many ways that researchers can generate their screening libraries, But current technologies leave much of the sequence space out of reach and underexplored. If your protein is longer than 300 nucleotides, your options for building screens are either very expensive, very limited, or very biased. As a result, the sequence space of most known protein sequences cannot be effectively studied in high throughput. To advance therapeutic protein development and AI-driven screening, researchers need tools to economically synthesize longer sequences at scale. At Twist, we constantly ask, how can we give more scientists access to longer synthetic DNA on a large scale? Our team used our proprietary silicon-based platform, optimized chemistry and automation to become the only company able to synthesize precisely designed pooled sequences that push the boundary beyond 300 nucleotides. This innovative new product, called multiplex gene fragments, consists of uniform pools of double-stranded DNA that are designed to your sequence specifications. In just 1 week, you can receive pooled lyophilized gene fragments, up to 500 base pairs ready for use in your high-throughput screen. With access to significantly more sequence space, you could precisely synthesize a library to generate training datasets to improve your machine learning model, or screen single-domain heavy-chain antibody variable regions and make discoveries that will lead to the next blockbuster protein therapeutic. So, what will you do with significantly more sequence space? Get started with multiplex gene fragments today.
Thank you everyone for that attention, and thank you Ryan for that incredible presentation. We're now going to move on to the Q&A session. Feel free to submit your questions using the box on the right hand side of the screen. Simply type them in and click send. We'll do our best to address as many questions as we can today. We've already had a few questions come right in, so Ryan, if it's all right with you, I think we'll dive straight in.
Please do.
Fantastic. So the first one we've had is, what distinguishes your lab in the loop system from other platforms being used for antibody optimization today?
That's a great question. And actually, I think a particularly great question to ask right after that video where we had the charming graphic of the hiker kind of ascending the mountain in several steps iteratively. I'd say the biggest thing that distinguishes our lab in the loop system is scale. So a lot of the way people have been thinking about this problem traditionally is as fundamentally an iterative problem where you are at a particular place, you screen as many variants as you can, you get a little bit up the hill, and you do that again three or four times. A lot of brain power, a lot of fantastic math has been devoted to kind of the active learning concept in ML, where we need to be really, really thoughtful about what data points we want to acquire next for model training. The big difference with our platform is we have, in some sense, a very simple solution to that, which is that during the first round, we simply generate 300 to 500 times more data than you're going to get by plating out proteins. And I think we've demonstrated at this point that with a wide enough look around you, you don't need to do it in multiple steps. So our typical MO, unless we're doing something incredibly difficult like the HIV work where we need to bind 1,000 different targets, is that our goal is to get enough of a view of the landscape the first time around that loop that we can simply ascend straight to the top. So I'd say our biggest difference there is we're really focused on keeping that funnel wider, longer, so that with enough data and the right amount of data, it doesn't need to be a difficult iterative problem.
Fantastic. As someone who enjoys hiking, I like that analogy. That was a good analogy there. So the next question we've had is, if you're focused on measuring affinity, how is it that with your approach, you can optimize all the other antibody properties in parallel?
Also an excellent question. And here I'd say we're really behind the thesis that affinity is fundamentally the problem. I like to make an analogy to kind of very traditional methods here. You know, when we think about antibody optimization, we normally think in a traditional sense about generating a hit, which has high affinity to its target, and then engineering it to be a good drug. And what I like to say is this seems normal to people, right? It's normal that you would take a hit that has good affinity, that you would make a few targeted edits, that you're looking for something that behaves better as protein that's more manufacturable. I think if you ask a traditional molecular biologist, they would say it would be absurd to take a really well-behaved antibody drug to some other target and think that you could make a few edits to make it bind your target. And what I think this calls out is just this basic fact about antibodies, that most antibodies aren't very manufacturable, but usually there's going to be some antibody within a few sequence edits of one that you start with that is going to be manufacturable. Whereas affinity really is the Wild West. It is basically impossible to find an antibody that specifically binds any given target that you'd like, other than doing some kind of massive primary screening. And so we think that space is really dominated, not by the fact that it's hard to find a manufacturable antibody. I really think if you're able to throw 1,000 or 10,000 darts, you'd find fantastic antibodies anywhere within the sequence space. It's dominated by the fact that the vast majority of antibodies are not going to bind your target, or even if you're starting with a hit, the vast majority of edited antibodies are going to lose that affinity to their primary target, which is a really fancy way of finally coming around to the question and saying, the way we do this is by mapping a sequence diverse space of really high affinity binders. We can then screen in silico for developability properties. And while that is imperfect, we generally get to a place that kind of within one plate of actual candidates that we make as protein, we consistently get hits that behave very well on the manufacturability scale. Though, of course, I'll call out that here we're kind of using the best available models for screening on those developability properties. And as continued advances accrue in that field, this is going to get even better. So effectively, what we do in a live campaign is we train our affinity model. We generate on the order of 1 million antibodies in silico that we're confident, 2/3 to 3/4 of those are still going to be as good or better binders than the parental, but they're really sequence diverse in primary sequence. And then we score them using in silico metrics, like the TAP suite from Oxford. We use natural antibody. There are a variety of techniques you can do to get things that are at least predicted to be biased in the right direction for expression, for melting temperature. We're starting to work more with kind of purpose-built sequence and structure-based models for specific properties like expression. But the answer is that basically we map out the space of high affinity antibodies, and then we really filter that down harshly based on predicted manufacturability phenotypes before deciding the small number that we're going to take forward into protein production.
Fantastic. Thanks for that. Oh, the next question we've had, someone said, I saw you have an existing database of 1.8 billion protein-protein interactions. Can partners or collaborators access that existing data?
Yeah, that's also a great question. I think the answer to that is a mix of get in touch. Obviously, not all of it. A lot of it's done with partners for particular therapeutic programs. But a pretty big chunk of it, is our data, and so there are ways we can make that work. And also, I'd say stay tuned, because we're really working on putting together a way to get our data out into the hands of the modeling community at scale, in a way that's more collaborative, more federated than just everyone getting their own data sets.
Fantastic. The next one we've had says, during the de novo design of mini binders, protein is often exclusively made of alpha helix. Could this be a problem?
That is a fantastic question. I have a short and hopefully not too unsatisfying answer here, which is yes, we do think that could be a problem. And a not insignificant amount of research time on our AI research team is dedicated to generating mini protein designs, which look a little bit more like natural folds from the human proteome. I'll also say that in some sense it depends on the application. There are certain contexts in which what you really want is a 3-helix bundle that is just an absolute rock and completely stable, and all you need to worry about is surface chemistry. Having said that, yeah, we have actually put a lot of thought into it, and we are confident at this point. The models can be some combination of nudged and filtered to generate folds that look a little more natural, even in this kind of smaller protein space.
Fantastic. The next one said is a bit broad here. What are your thoughts on predicting antibody binding and developability using linear sequence-based models versus structure-based predictions?
Fantastic question. With the caveat that this is my personal opinion, so please don't judge me too harshly in 5 years. My thoughts, and this is kind of somewhat informed by a lot of the things that we've been doing, including AlphaBind and beyond, is that sequence-based models are absolutely spectacular within a narrow space. So AlphaBind for antibody optimization is purely sequence-based, and it absolutely knocks it out of the park. It's everything you would want in a fine-tuned affinity model. We think generalization is much harder for sequence-based models, particularly on the target side. We think the key constraint here is target diversity. And so again, my personal opinion is that when we think about how do we get models to effectively generalize to new targets in a zero-shot context, where now we want to really boost performance, maybe even without having candidate-specific fine-tuning data, that's something where we really have to go through a well-informed structural intermediary.
Fantastic. We've had two questions come in that are a little bit more speculative in terms of applications. So he said, do you have any experience with expression of antibodies in bacterial systems?
Yeah, that's a great question. So I'd say, limited. We've tried various things as a research lab in molecular biology. I will say a large majority of our current work is in CHO expression systems.
And the other one said, are you looking at protein kinase inhibitors to maximize their sequences to optimize binding?
Yeah, I'll say there's probably not much I can say about what specific drugs we or others are working on. I will say I don't think we've announced anything publicly in kinase inhibitors. To a first approximation, about half to 2/3 of what we do is antibody work with some non-antibody biologics, and then we do a lot of work actually in targeted protein degradation, where we're kind of doing proteome level scans for novel latent interactions. But we do think there are opportunities kind of across the proteome for any time we want to optimize a protein-protein interaction.
Fantastic. We've got a few minutes left, and we've got one question here that is quite, I think, a good big picture question. Again, we won't hold you to any of your predictions, but what are your thoughts on how wet lab and dry lab integration will evolve over the next few years?
Yeah, that's a great question. Super open-ended, but something that's really important. So, I will go out on a limb and say I think we will see the total ascendance and victory of lab in the loop as a philosophy, simply meaning that the need to capture, integrate, and meaningfully use all of the data that's coming kind of downstream of the actual protein AI models is increasingly recognized, I think, across the industry. And in some sense, as labs are getting more automated, as kind of LIMS and ELN systems are getting more sophisticated, I do think it's getting much easier than it used to be to get all of this data in one place and use it. So, that's one thing that I think is important, is that people are going to be, I would like to think that 10 years from now, we get to a place where, almost every piece of data that's being generated in the lab is kind of appropriately documented and sent to the right place, whether that's, validating that models are working, whether that's just general operational health metrics, whether that's feeding into the hopper for further model retraining and refinement. And the other thing I'll say is that I do expect kind of continued development on the technology front and a lot of synergy between wet lab and dry lab systems. So I think today's talk was a great example of those synergies. Without modern AI protein design systems, there'd be no call to build DNA, with dozens of edits across an antibody sequence. It would be pointless. You'd never get anything out of that. But without the ability to generate those data at scale, we cannot effectively use those AI models. And so they really do go hand in hand. I'll also say that, there are a lot of constraints that people get used to very quickly, but that aren't kind of real constraints. They're constraints based on current technology. So as we talked about, there's been a lot of work out on iterative lab in the loop systems where we do a lot of active learning, all based on this fundamental axiom that it's never going to be cheap or easy to get lab measurements at the scale that we'd actually like for AI model training. So we're going to have to live in a world where we're stuck with very, very small batch sizes, and we have to be extremely thoughtful about what data to acquire. And hopefully I've shown today that turns out not to be a real constraint. That's a constraint purely based on the technologies available in the wet lab. And as new wet lab technologies come out, right, it suddenly becomes possible to do things that didn't seem possible before, like simply gather enough data to really solve your particular problem in one go. And again, likewise, as better models come out, things that had previously been very difficult now become plausible. So as I said, we're really excited about a lot of the recent papers in de novo antibody design recently, which have just gotten to the point where it's possible to send a plate or two of candidates and actually expect to get some hits out, which we think is a really, really exciting inflection point for routine use of some of these models. And I'll end with, the more we do big data, the more we can do with AI, and the more we can feed that data back into AI systems. So one example that we love is that, effectively every time you do de novo design, you have a structure that you think is the bound structure of your candidate to the target. And when that works in the lab, it has been an empirical observation, and there's good reason to believe this, that when it works in the lab, probably the co-structure is almost correct. I believe atomically accurate is the term of art in the papers. And one cool thing this means is that every time you successfully validate a de novo design, you've got a data point that is maybe not as good as a crystal structure, but in the same ballpark as a crystal structure. And so as we start to accumulate those, we think the ability for big wet lab technologies to validate at scale will generate more training data, which will itself accelerate the growth in performance of these AI models. And so we think getting that flywheel going, where the AI is powerful enough to use at scale, cheap enough to use at scale too, and then that we've got wet lab technologies that can validate at scale, can actually potentially lead to the kind of exponential growth in data and consistent improvements in model performance that we've seen in other fields and that we'd really love to kick off for protein engineering.
Fantastic. That seems like a really optimistic note to end on, Ryan. So we're now going to wrap up. Again, big thank you to everyone for everyone's attention. Please remember that any questions we didn't get to will be answered offline as soon as possible. You can also continue to ask questions even when watching this webinar on demand. Don't forget, the certificate of attendance will be sent to you via email once the webinar closes. Once again, a big thank you for your attention and a huge thank you to Ryan. We really appreciate your time today, particularly in answering the questions. Thank you very much. Goodbye.